I'm George Hammond, Senior Conference Producer at Technology Networks, and I'm here to introduce our next talk, Digital Twins in Vision Assessment, a novel approach for predicting the contrast sensitivity function. It is my pleasure to introduce Dr. Zhong-Lin Lu. A renowned neuroscientist and psychologist, Dr. Lu is a professor of neuroscience at NYU Shanghai and a global network professor at NYU. He earned his PhD in physics from NYU and transitioned into cognitive neuroscience during a postdoctoral fellowship at UC Irvine. Dr. Lu has held prestigious roles including the William M. Keck Chair in Cognitive Neuroscience at USC and Director of the Center for Cognitive and Behavioral Brain Imaging at OSU. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I'd like to hand over to Dr. Lu to begin his presentation.
Thank you, George, for your introduction. Before my presentation, I must disclose that I have personal financial and intellectual property interests in Adaptive Sensory Technology, Inc. and Jiangsu Juehua Medical Technology Limited. Mary has just been diagnosed with a serious eye disease that requires immediate treatment. The doctor told her that the standard treatment isn't very effective, but there is an ongoing randomized controlled trial for a new drug that shows promise and could potentially be highly effective in treating her condition. If she agrees to join the trial, she will be randomly assigned to one of two groups. There's a 50% chance she will receive the experimental drug and a 50% chance that she will receive a placebo. The trial lasts 5 years. To complicate matters further, there's a possibility that a different treatment, one that could also help her condition, might become available halfway through the trial. Mary now faces two choices. First, participate in a clinical trial, knowing that there's a 50% chance of receiving the experimental drug, but there's also a 50% chance of receiving a placebo. Number two, wait for the other potential treatment to become available in the future. What should she do? To minimize biases, randomized controlled trials randomly assign subjects to an experimental group and one or more control groups. In some randomized controlled trials, known as placebo-controlled trials, at least one control group receives an inactive substance or placebo instead of the treatment under investigation. The goal of a placebo-controlled study is to determine whether an intervention is more effective than a placebo. In active control trials, the control group receives a currently accepted intervention, and the experimental group receives a new intervention. The goal of an active control trial is to determine whether the new intervention is more effective than the current ones. Some trials compare the new treatment to both the placebo and the accepted treatment. Because a current intervention is likely to have a significant effect on human health, whereas a placebo probably will not, comparative trials or active control trials usually require larger sample size than placebo-controlled studies because the difference between the new intervention and the current one may not be very large. Although many clinical researchers and biostatisticians regard placebo-controlled randomized controlled trials as methodologically superior to comparative trials, placebo-controlled randomized trials in clinical research are ethically controversial because the subjects in the placebo group may be denied an effective therapy. In some cases, subjects in the placebo group may be denied medical care. Many codes and guidelines state that it's unethical to withhold a proven effective therapy from research subjects with a serious medical condition. In this talk, instead of a debate on the ethical issues in randomized controlled trials, our research question is, even if we must conduct placebo-controlled trials, can we reduce the sample size of the control group? Both U.S. Food and Drug Administration and the European Medicines Agency have issued guidelines for using digital twins in randomized controlled trials. These are called TwinRCTs. Digital twins of patients in a treatment arm, can be used as virtual patients in a control arm to increase the effective number of patients through combination with real patients and can enable faster and smaller trials by enhancing statistical power. Digital twins have been utilized for safety assessment in over 500 FDA submissions. The digital twin is an ideal framework for product lifecycle management. It includes a physical system, a corresponding virtual model, as well as a bidirectional data flow that provides physical to virtual and virtual to physical connections. The American Institute of Aeronautics and Astronautics extended the definition of digital twins. It's a set of virtual information constructs that mimics the structure, context, and behavior of a natural, engineered, or social system or system of systems. It's dynamically updated with data from its physical twin, has a predictive capacity, and informs decisions that realize value. The bidirectional interaction between the virtual and the physical is central to the digital twin. Digital twins have found numerous applications in fields where forecasts and predictions are crucial. This includes climate sciences, business, engineering, finance, and healthcare. According to McKinsey and Company, investments in digital twins will surpass $48 billion by 2026. In healthcare, proponents of precision medicine suggest that digital twins can help identify individuals at risk for disease, which provides the opportunity for early intervention to prevent worse outcomes. The prediction of treatment outcomes can also enable the development of personalized interventions optimized for each individual patient. And also, as I mentioned earlier, both the FDA and EMA have issued guidelines for using digital twins in TwinRCTs. The scale and modeling methods of digital twins depend on the nature of the physical system and the desired level of detail, fidelity, and functionality. In healthcare applications, digital twins can span a broad spectrum of biological scales, encompassing molecular, subcellular, and cellular levels, as well as entire systems, functions, individual humans, populations, and the biosphere. They can also represent medical devices, such as the OCT machine or healthcare organizations. Various digital twin modeling methods have been developed to create sufficiently representative virtual replicas of physical entities, processes, or objects, including geometric modeling, physical-based modeling, data-driven modeling, physics-informed machine learning modeling, and system modeling. In this study, we employed a data-driven generative model approach to develop digital twins for the contrast sensitivity function based on a group of human observers tested under different luminance conditions. The contrast sensitivity function characterizes contrast sensitivity as a function of spatial frequency, illustrated by the white curve in this graph. The background of the graph shows sine wave gratings with increasing spatial frequency from left to right and decreasing contrast from bottom to top. The white curve separates the space of sine waves gratings into visible and invisible regions. As a fundamental assay of spatial vision, contrast sensitivity function is closely related to daily visual activities in both normal and impaired vision. It has emerged as an important endpoint for staging eye disease and assessing treatment efficacy. Importantly, the CSF varies not only with stimulus conditions such as retinal luminance, temporal frequency, and eccentricity, but also with disease progression and treatment. Predicting CSF for new observers or existing observers in not yet measured conditions could help predict human performance in new conditions, identify potential risks and benefits of interventions, and enable personalized treatment for each individual patient. The predictions could also serve as informative priors to reduce test burdens in new measurements. At the group level, the digital twins of clinical study patients in active arm have the potential to enable TwinRCTs. The FDA currently considers multiple measures of visual function as acceptable primary endpoints for clinical trials that evaluate the safety and effectiveness of drugs, including but not limited to contrast sensitivity, provide there are clinically and statistically significant differences in multiple spatial frequencies. If the contrast sensitivity function is so good, why doesn't everybody use it? It's because of prohibitive test time. To measure contrast sensitivity function requires measurements of contrast thresholds at 5 to 6 spatial frequencies with a minimum of 30 to 50 trials per frequency, and a total of 200 to 300 trials or 20 to 30 minutes. This is just for one eye in one test condition. If you consider humans have two eyes, we have two monocular tests and binocular vision and multiple conditions, the test time is prohibitive. There have been various shortcuts for contrast sensitivity tests. This includes many different charts like Patty-Robson chart, mask chart, vector vision CSV 1000 chart, and FACT chart. These charts have some serious sampling limitations because they only sample this contrast spatial frequency space very coarsely at only limited spatial frequencies and or limited contrast levels. To measure contrast sensitivity function accurately and precisely requires high resolution sampling, as illustrated in this figure. As you can see in this figure, if we provide very high sampling density, the issue is that the test time could go up very, very fast. The solution is efficient sampling. If we can sample stimulus only around the contrast sensitivity function itself, we could provide very precise measurements, at the same time reduce excuse me, the test time. About 15 years ago, our group invented the quantitative contrast sensitivity test. In this test, we characterized the CSF with 4 parameters: peak sensitivity, peak spatial frequency, bandwidth a half width, and low spatial frequency truncation. We developed an active learning algorithm by searching a library of potential contrast test items. Then we test with personalized, optimized items in each trial. Then analyze responses with a computational model. Then we repeat until they converge on a base optimal test sequence. So this algorithm allows us to reduce the test time of contrast sensitivity function by about 10 times. Here's a demonstration of the algorithm. In this graph, the black curve is the underlying contrast sensitivity function. The green curve is the current prior of the contrast sensitivity function before we conduct any trial. These other graphs are posterior distributions of the parameters of the contrast sensitivity function. As trial goes on, you can see that each dot in this graph represents a trial. A blue dot means the subject made the correct response. A red dot means the subject made the incorrect response. As trial goes on, the green curve approaches the black curve, providing an accurate and precise measurement. of the contrast sensitivity function. Later, we further improved the contrast sensitivity function test by using bandpass filtered letters. With this 10 alternative fourth choice task, we further improved the speed by a factor of 2. At this point, we can obtain a very precise and accurate measurement of the contrast sensitivity function in about 2 to 5 minutes. So this is a new depiction of the contrast sensitivity function. It basically divides letters that are visible with large size and high contrast from letters that are of small sizes and lower contrasts. The application of the contrast sensitive quick CSF test have found a lot of applications in clinical research. One major findings in a lot of these studies is that patients in the early stages of ocular disorders may experience deficits in contrast sensitivity function even when visual acuity remains normal. In fact, subjective visual complaints are common in ophthalmologic clinics, even among patients with 20/20 visual acuity. In this graph, I'm showing you data from a number of diseases, including neovascular age-related macular degeneration, retinal vein occlusion, intermediate AMD, geographic atrophy, non-prolific diabetic retinopathy, and diabetic edema. All these patients demonstrate good visual acuity, but deficits in contrast sensitivity in the intermediate and high spatial frequencies. Previously, we developed a 3-level hierarchical Bayesian model to comprehensively model entire CSF dataset within subject design. This model utilizes trial-by-trial data and employed a log proper CSF functional form with 3 parameters as a generative model at the test level. In addition to hyperparameters at the subject and population levels, the model incorporates between and within subject covariances, as well as conditional dependencies across levels. The performance of the HBM was evaluated using an existing dataset of 112 subjects tested with quantitative contrast sensitivity function across 3 luminous conditions. And the model improved the precision of the QCSF results by a factor of 2. Importantly, it recovered many important covariances at all 3 levels. Here, we expanded the application of the HBM to generate digital twins for population of CSF observers. The trial-by-trial responses from historical data were used to train a HBM model. This model derives the joint posterior distribution of CSF hyperparameters and parameters at the population, subject, and test level. It incorporates conditional dependencies across the 3 levels to improve estimates of CSF parameters for each test. In the HBM, the population level distributions of hyperparameters describes the probability of CSF parameters across all the subjects. The subject level distributions, which are conditionally dependent on the population level hyperparameters, represents the probability of CSF parameters at the test level. These parameters determine the trial-by-trial response probabilities in each CSF test using a log proper model. The generative model allows the digital twins to combine the joint posterior distribution with newly acquired data to generate predictions for observers in unmeasured conditions. These predictions can also serve as informative priors for subsequent testing in those conditions. Our hypothesis is that the digital twins created using the HBM can make accurate and relatively precise predictions of CSFs for new or existing observers in conditions where data is not available. To test this hypothesis, we conducted and assessed 12 prediction tasks using an existing CSF dataset involving 112 subjects. We divide the subjects into two groups. Group 1's data in all three luminance conditions are treated as historical data. For group 2 subjects in the 12 prediction tasks, in task 1 to 3, there's no new data in group 2, but we try to predict this group 2's performance in each of the three luminance conditions. In prediction task 4 to 9, we know something about group 2. We have lab data in only one of the luminance conditions. We try to predict their performance in the other two luminance conditions. In task 10 to 12, we know the performance of new group 2 in two luminance conditions. We try to predict their performance in the third luminance condition. First, we discovered correlations in historical data. At the population level, the HBM find strong positive correlations between peak gains across luminance conditions and strong negative correlations between peak spatial frequency and bandwidth within each luminance condition. At the subject level, it identifies strong negative correlations between peak gain and peak frequency and strong negative correlations between peak frequency and bandwidth within each luminance condition. Additionally, at the test level, the model finds strong negative correlations between peak frequency and bandwidth within each luminance condition. So this slide shows individual subject level predictions. In column 1, we're showing the actual data from this observer in the low luminance condition. In column 2, we have no new data for this subject. We try to predict the subject performance in low luminance condition. In column 3 and 4, we know the subject's performance in the medium and high luminance conditions. And in the last column, we know the subject performance in both medium and high luminance conditions. Similar patterns were observed in those in the M and H conditions. We run a linear discriminant analysis. We find that the distributions from the predictions and the observed are not discriminable. This figure displays scatter plots of the predicted and observed area under log CSF for all subjects in the 12 prediction tasks. Across all prediction tasks for all 56 subjects, none of the predicted CSF parameter distributions were significantly different from the observed. The average standard deviation, however, of the predictions were about 3 to 2 times of those from the observed data. This graph shows the group level predictions. To construct this group level predictions, we average the predictions of all the 56 subjects. In column 1, again, this is the group average performance in the low luminance condition. In the second column is the group average when we have no information for any of the subjects in this group. In column 3 and 4 is when we have data on the medium and high luminance conditions for this group. And in the last column, we only have data for this group in both medium and high luminance conditions. We try to predict low luminance condition. Similar patterns were observed when we try to predict the group performance in the medium and high luminance conditions. None of the predicted CSF parameter distributions at group level are discriminable from the observed, like this observed distributions. Again, the standard deviation of the predict CSF at the group level are about 2 to 3 times of those of the observed. This suggests where the predictions were quite accurate. The predicted AOL CSF has lower precision. If we consider the M condition as the baseline and the H and L conditions as the treatment and control arms in an RCT, predicting performance in the L condition with new data in both the M and H conditions, namely task 12, effectively emulates a TwinRCT. In this setup, digital twin predictions for the 56 group 2 subjects in the L condition The control arm will derive from their baseline, the M and treatment H data. And despite a lower precision in quantifying those virtual patients in digital twins, their inclusion can still notably increase the effective sample size in the control arm, thereby enhancing the statistical power of the twin RCT. We can also use the predictions of the digital twins to improve the Bayesian active learning system for QCSF. The test is based on a Bayesian active learning algorithm. It uses information acquired during the experiment with a prior knowledge about the CSF's general functional form. Before any data is collected, the initial prior is used to represent foreknowledge of observed CSF parameters. Informative priors could further improve the QCSF test. We evaluate the benefit of using the predicted parameter distributions in the 12 prediction tasks as informative priors in the QCSF procedure. This figure depicts the distribution of the number of trials needed to achieve statistically equivalent precision as observed AOL CSF collected by 25 QCSF trials. On average, only 11 trials were necessary. We also determine the number of trials needed to obtain estimate AOL CSF that are not statistically different from the observed data and find that about 9 trials on average were required to achieve the same accuracy. Combining these results together, we concluded that informative priors can save about 14 trials or 56% of the data collection burden in QCSF. In summary, we developed and validated digital twins for population of contrast sensitivity observers using a data-driven generative model approach based on a 3-level HBM that captured both between and within subject covariances. The digital twins demonstrate high accuracy in group-level predictions across all tasks and maintain accuracy at the individual subject level when new data were available. The key innovations are the development and application of HBM-based digital twins in the functional vision domain, the novel approach of treating prediction tasks as missing data problems within the HBM framework, the incorporation of digital twin predictions as informative priors in QCSF testing. In ongoing research, we're making development of digital twins to incorporate both functional and structural vision assessments, as well as development of digital twins for predicting disease progression or treatment outcomes. In the laboratory of the future, digital twins will revolutionize the way we understand and improve systems. By harnessing historical data and leveraging the power of machine learning, we can build models that uncover the most critical relationships within the data. These models become the digital twins of the original systems, providing a dynamic, evolving representation of reality. As we gather new data, we can feed it into these models to make insightful predictions, guiding targeted interventions that improve outcomes. The laboratory of the future is no longer just a place for static data collection. It's a vibrant, adaptive environment that continuously learns and evolves, growing ever more efficient. And as a cornerstone of personalized medicine, it holds the promise of more precise, effective, and tailored solutions for patient care. Thank you very much for listening to my talk. I would also like to acknowledge all my collaborators, Yukai Zhao, Lu Nesmus, Michael Dorr, as well as the support of NSF, NIMH, and NEI for the work presented today. Thank you for your attention. Now I open the floor for questions.
Thank you very much, Dr. Lu, for that incredibly fascinating presentation. We really do appreciate you sharing your expertise and your insights with our audience today. Now, as with all the other talks, we're likely going to have lots of questions come in from the audience, and I can see that we have received some questions already, so I'll move straight onto the Q&A now. So, Dr. Lu, your first question is, How does the hierarchical Bayesian model enhance the accuracy and reliability of predictions in this study?
The HBM actually has three different levels. It has hyperparameters, the population subject levels, and parameters, the test level. And it also incorporates covariances between subjects at the population level and within subjects at subject level and test levels. So it's this conditional dependencies that uses information across subjects and the covariances that quantify the relationship between the parameters across all the tests that enable the HBM to make those predictions.
That's a fantastic answer. Thank you so much for that, Dr. Lu. Now, moving on to your next question today, how can the approach used in this study be adapted for other visual metrics beyond contrast sensitivity function?
So the most important aspect of the prediction is the generative model. In this case, we have a three-parameter generative model, the Slug proper model of the CSF. Once we acquired a generative model, for example, for visual acuity, for example, for OCT measurements, we could use exactly the same framework to extend the applications in those domains.
That's excellent. Thank you so much. We do have time for a few more questions today before we close out this session, but just a reminder to the audience, any questions that we don't have time to get to will be sent to the speaker to be addressed offline as soon as the event closes. Now, moving on, Dr. Lu, what are the limitations of this study regarding the generalizability of the digital twin model to other populations or conditions?
The limitations of this current study is that it's a simulation study based on data collected from college students. We emulated the visual deficits using banked foil filters. Oh, sorry, in this case, we used the goggles to emulate with different luminance conditions to emulate visual conditions. And to make this digital twin technology more useful, we need to have clinical data of real patients ideally over time. So we could actually not only make predictions about their performance in different conditions, but also make predictions in time of their progression or their treatment outcomes.
That's another excellent answer. Thank you so much, Dr. Lu. Now, I have no doubts that this study will have many implications on the real world, but in your opinion, what implications do the study's findings have specifically for clinical practice in vision assessment and for treatment planning?
I think this requires it has huge implications for treatment and assessment in the sense that if we have historical data of a lot of patients in each one of the eye diseases, we could actually use the historical data to actually make predictions of disease progression and treatment outcomes for each individual. Imagine you have a new patient who come to your clinic. You want to make a plan for the new patient's treatment. What you want to know is how each treatment, like different treatment options or treatment regimes could function for this person. You can actually use digital twins to help you to select the best treatment plan for the patient on the first day. And of course, as time goes on, you can reevaluate the treatment in the next visit, and you can figure out based on the data you have, the historical data and the new data you have from the patient, you could actually make a better prediction about the treatment outcomes of different treatment options. Then you choose the optimal one based on that, and so on and so forth. On the assessment part, as I showed in this talk, that with the predictions from digital twins, we can significantly reduce the data collection burden. In this case, I showed you that we can reduce the time of test by about 56%, which is highly significant in clinical trials and also in the clinic, as we know that our clinicians really don't have a lot of time with each patient. If we can reduce the test time, that reduces the burden for our clinicians and also for our patients.
That's great. Thank you so much, Dr. Lu, and incredibly fascinating. I can't wait to see how that translates. Now, we have time to go through one final question before we close out the session today. So, Dr. Lu, your last question is, in what ways can the findings of this study be applied to broader contexts, such as precision medicine, or in the case of twin RCTs?
So I think this is a conceptual study, as I said, that we've been trying to develop a technology to enable twin RCT. And we have a demonstration case in which we show that the predictions of the digital twins are accurate. And even though it's less precise, if you include the virtual patients, even with less precision, could significantly reduce uh, the number of patients required. So in fact, in this study, there are two aspects that I showed in the twin RCTs. First is reduction of number of subjects in the control arm, which addresses the question I stated in the very beginning, which is, in placebo-controlled clinic randomized control trials, the ethical issues of denying patient treatment is uh, is huge. We could reduce that part, reduce that kind of ethical problems, number one. Number two, because we can improve the efficiency of the tests, we could significantly reduce the test burden. And this translates into huge benefits in terms of test time and money savings and a lot of those things. And more importantly, improves the precision of the tests and make it more effective, make the randomized control trials much more effective.
Excellent. Thank you so much, Dr. Lu. Unfortunately, that is all that we have time for today. As mentioned, any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. So that just leaves me to say a massive thank you once again, Dr. Lu. We really do appreciate you sharing your expertise and your insight with us today. And to everyone in the audience, we'll be back soon with the next talk. Goodbye.
Thank you.